And there's three basic sections of the collecting duct to know, and they're easy. One is the CCD, that's the cortical collecting duct. That's the collecting duct in the cortex of the kidney. Then an OMCD, an outer medullary collecting duct. That's the collecting duct in the outer medulla. And then an IMCD, or the inner medullary collecting duct, the collecting duct in the inner medulla. Okay? So you need to know the basic structure of the kidney, gross anatomy, cortex, and medulla. Medulla can be divided into an outer medulla and an inner medulla, and then know the basic nephron segments from the renal corpuscle. This is Bowman space that I'll go into in just a second. Proximal convoluted tubule, proximal straight tubule, descending thin limb, ascending thin limb, thick ascending limb, distal convoluted tubule, cortical collecting duct, outer medullary collecting duct, inner medullary collecting duct. Different cell types all along these nephron segments, even different cell types within a nephron segment. So that basic anatomy should tell you something about the function of the kidney, which is what? There's lots and lots of functions, lots of different cell types with different transporters, different functions, so the kidneys are doing a lot of things. Okay, and that's what we'll talk about, maintaining sodium balance, calcium balance, potassium balance, magnesium balance. And they have to transport glucose. They have to maintain the osmolarity of the body fluids. Okay, we talked about osmolarity yesterday in body fluids. The kidneys are responsible for keeping our plasma osmolarity constant and normal. The kidneys are responsible for keeping the blood volume constant and normal. So the kidneys are responsible for keeping the blood pressure constant and normal. So all of these functions the kidneys have to do are served by all of these different cell types that the kidneys have. Okay, so that's the basic structure of the kidney. Go into the cellular features of the nephron segments. I think you know this from histology already, but let me just let you know what you need to know about this. Proximal tubule, okay, you should know that, remember from histology, these cells have what's called a brush border on their luminal side. Okay, and we'll review this again in the lessons to come about apical versus basolateral and transport, reabsorption, secretion. But the proximal convoluted tubules are pretty big. Know that the luminal side has a brush border, which increases greatly the surface area for a lot of transporters and a lot of things to do, just like a brush, it's kind of bristled in there. And they are abundant with mitochondria. And you know what mitochondria do. Right? Oxidative phosphorylation, generating ATP, constantly working. Of all of the cells in the body, perhaps the proximal tubular cells are the most metabolically active because of all of the things that they're doing. They handle all of this filtrate in a bulk way. So that's the morphology of the proximal tubule. As we go down the descending thin limb, they become thin, not very much mitochondria. There is some important permeability characteristics that we'll talk about later. Thin ascending limb cells, or ascending thin limb cells, are also thin, obviously, not very much mitochondria. Not much transport takes place in there, but it's been hard to study. We come up into the thick ascending limb, and these do have abundant mitochondria. They don't have the brush border like the proximal tubule cells have, but they do have a very important transport function. And we'll talk about that later. So thick ascending limb cells obviously are thick with abundant mitochondria. Same thing in the distal tubule. Okay, and they actually have a different type of transport function that becomes important, but they're similar to the thick ascending limb cells. Now the macula densa cells I'll talk about in a second. These are specialized cells in the thick ascending limb. And notice here, in each and every nephron, this is what happens. The thick ascending limb cells come right up very close to the microcirculation of the kidney. This might be an afferent arteriole. Here are your glomerular capillaries. Here's your efferent arteriole. And these macula densa cells actually touch these arterioles. Okay. So that's a very important function. That's part of the juxtaglomerular apparatus that I want you to know about. So three parts of the JGA: macula densa, granular cells of the afferent arteriole that secrete renin, and these so-called extraglomerular mesangial cells that I'll talk about in a second too. So the distal tubule, and then we come into the, from the distal convoluted tubule, you might also hear of a connecting segment between the distal convoluted tubule and the cortical collecting duct. That connecting segment is sometimes shown and sometimes it's not. What I normally refer to as an early distal tubule and a late distal tubule. So the late distal tubule actually represents more of the cortical collecting duct cells. Okay, in the collecting duct we have principal cells and intercalated cells. Okay, so know the names of those two cells and where they exist. We'll talk about their function later. Then we come into the outer medullary collecting duct and then the intermedullary collecting duct, and the IMCD cells appear to be quite uniform, and they do have a moderate amount of mitochondria, but they also serve an important function. Okay? So for right now, just be able to list those nephron segments, each one of them, and get the basic morphology down of each cell. Okay? Then when we talk about the function and all of these things the kidneys will do, should make a little more sense to you. Juxtaglomerular apparatus, as I showed earlier,